My name is Jessica Sable, and I'll be presenting the genomic analysis of RCCX gene cluster in Homo sapiens. I'll be discussing the disease background, connection to genomics, genomic analysis, and the summary. So first, some background. The RCCX gene cluster is located on chromosome 6 within the major histocompatibility complex class 3 region. The cluster contains 4 main genes: C4, TNXB, CYP21A2, and RP1. C4 is broken into two parts, C4A and C4B. The RP1 is classified both as a gene and a pseudogene depending on the context. And the RCCX locus also contains pseudogenes RP2, TNXA, and CYP21AP. So here's the structure of the RCCX locus, containing 2 segments and spanning centromere to telomere. The arrows indicate the orientation of the open reading frames of the genes. The RCCX region is prone to different mutations as well as formation of chimeric genes, and the genes in the cluster are close to each other and have the tendency to go through copy number variations, which causes the cluster to behave as a unit and go through mutations together. This diagram illustrates copy number variation, which are monomodular, bimodular, and trimodular as the 3 types. They either involve a single copy of all genes, 2 copies, or 3. Bimodular is the most common configuration in humans, but monomodular or trimodular configurations can make a human more susceptible to disease. The formation of gametes during meiosis may cause the RCCX loci variants to be produced, which contain non-coding repeats. The locus also contains repetitive elements that can lead to variations in the genome structure. This is important because the genetic rearrangements are associated with having an increased risk of developing a genetic disorder. There are disease states associated with each gene in the cluster. C4A is associated with diabetes type 1 and systemic lupus erythematosus. C4B is also associated with lupus, and SLE is driven by C4A deficiency primarily rather than C4B. TNXB is associated with hypermobile Ehlers-Danlos syndrome, and the TNXA-TNXB chimera is associated with congenital adrenal hypoplasia X syndrome. CYP21A2 is associated with CAH, postural orthostatic tachycardia syndrome, and mast cell activation syndrome. Here's an illustration showing some of the genetic changes that occur that lead to a disease. C4 experiences reduced copy number, CYP21A2 experiences various mutations, and TNXB experiences mutations and deletions. Here are examples of genetic changes that occur in the RCCX cluster and how they clinically present. In example A, the CAH chimera is caused by a recombination between CYP21AP and CYP21A2. This causes the impairment of CYP21A2 and the deletion of C4P, but leaves the TNXB gene safe. This results in CAH. In example B, the CAHX chimera is produced by recombination between TNXA and TNXB and produces the complete deletion of CYP21A2 gene, the impairment of TNXB gene, and the deletion of C4B, resulting in CAHX syndrome. In example C, C4CNV leads to a high C4B copy number, which is an Alzheimer's disease risk factor, and the C4A deficiency is related to SLE. The RCCX gene theory was first proposed in 2015, and it states that because the genes in the RCCX gene cluster act together, many people experience overlapping syndrome. This supports the idea that variation in the gene cluster lead to a predisposition of chronic illness. The hypothesis states that both copy number variations and polymorphisms in the RCCX gene cluster lead to a predisposition to autoimmune connective tissue and adrenal disorders. The RCCX gene cluster is significant for many reasons. It is structurally dynamic and genetically complex. The C4 genes are involved in immune function, and the cluster contains both genes and pseudogenes. Because of the structure of the locus, it is a great model for studying genetic instability. Why use genomics? So genomics is used to visualize the entire genome of any given organism. Genomics is used in this analysis to see how genomic regions, as opposed to just individual genes, influence disease states. To perform this genomic analysis, different software were used. This includes the UCSC Genome Browser, the NCBI database, and NBLAST, the GWAS catalog, and the GTEX portal. In the UCSC Genome Browser, I searched the MHC class 3 and zoomed out 10x twice. This shows all the RCCX genes in the cluster as well as additional pseudogenes. In the ClinGen copy number variation pathogenic track, there is a large blue bar spanning from pseudogene STK19 to TNXB. This indicates a pathogenic copy number gain in all 4 genes in the RCCX locus. These results indicate that the RCCX gene cluster is susceptible to structural variation, and a gain affecting these 4 genes is clinically significant due to being potentially pathogenic. Because the gain is covering all 4 genes in the RCCX locus, this suggests a bimodular or trimodular phenotype. Here is a continuation of the results showing gene expression in different tissues as well as the alignments of 100 vertebrates. This is another continuation of the results showing pseudogenes and their location. To validate pseudogenes that have been previously identified, a BLAST search was performed aligning CYP21A2 and TNXB. Here is the input for the NBLAST. The BLAST showed 1 hit on chromosome 6, and when looking at the features of the hit, we see a chimera of CYP21A2 and CYP21AP. To validate polymorphisms, the GWAS catalog and the GTEX portal were used. In C4A and the GWAS catalog, there were 4 associated variants identified, all with different traits. The C4A bulk tissue gene expression showed high expression in the liver, ovary, and adrenal gland. In C4B, the polymorphism showed 3 hits, all significant to SLE. C4B also has the highest expression in the adrenal gland. In lupus, the most significant organs are thyroid, parathyroid, thymus, pituitary, adrenals, pancreas, ovaries, and pineal gland. This explains why the results also show expression in these tracts. CYP21A2 showed hits for 58 variants. The ones of most significance were variants associated with the C4 complement. CYP21A2 showed the highest expression in the adrenal gland, which supports the fact that this gene is responsible for adrenal hormone synthesis. TNXB showed 13 variants, with 3 significant for autism and schizophrenia. These conditions have been proposed to be associated with the RCCX gene theory. And in TNXB, there were other variants, with multiple being C4 complements. The bulk tissue expression for TNXB showed high expression in multiple tracts. Of significance, the highest was in esophagus and adipose tissue. Polymorphisms were also investigated using NCBI. In TNXB, there were multiple hits showing intron variants. One of the variants from the previous slide, RS6460, was performed with a BLAST to get different perspectives on polymorphisms. And these were the results. To compare species that have the RCCX gene cluster, a BLAST was performed. The RefSec annotation NM019108-105.8 was used to compare TNXB to the rhesus monkey. And NBLAST was used for this comparison. The results of the BLAST showed predicted sequences with the rhesus monkey. Another comparison was done using C4B with the RefSec annotation NM001002029.4 being used for the BLAST. C4 was used in a BLAST to compare with chimpanzees. The results showed 2 hits for chimps, and this was greater than 99% identity. In summary, the pathogenic CNVs in the locus suggest disease potential in all 4 genes. The SNPs relating to C4 TNXB and CYP21A2 show traits associated with the diseases listed in the RCCX gene theory, as well as SLE and CAH. The rhesus monkey and chimpanzee show significant hits for genes in the cluster, and this gene cluster helps for better understanding of the connection between certain chronic condition. This could be used in humans, allowing for genetic testing to help them get a diagnosis and potential treatment. And here are the references I used to complete the analysis. Thank you.